My name is Kamal, and I'm from Department of Plant Pathology, NDSU, and I'm with Dr. Guiping. And today, I'm going to talk about impacts of different cover crops on hatching, penetration, and development of soybean cyst nematode. So let's start with soybean, and we know that I don't have to talk about soybean a lot because we know a lot about soybean, and a previous speaker has already talked about it. But nonetheless, soybean is a very important leguminous crop because of the higher oil and protein content in it, and a lot of stuff that we use in our daily life and other sectors are derived from soybean and its byproducts. So USA ranks second for the production of soybean in the world with the value of more than $31 billion. There are a lot of biotic and abiotic problems in the soybean production, and among the biotic factors, soybean cyst nematode is the biggest threat to soybean production in the USA as well as other soybean producing countries. Here in the slide, you are seeing on the left-hand side of your left-hand side, you are seeing the cyst of soybean cyst nematode. There are brown cysts as well as some of the white cysts as well, depending upon their maturity. The darker the color, the older the cysts are. And on the right-hand side, you are seeing soybean cyst nematode eggs and juveniles. These juveniles are the second stage juvenile, and they are the infective stage of this nematode, and they infect the suitable host and penetrate their roots. So let's talk about soybean disease cycle in a brief. The disease cycle starts with the hatched juvenile, J2, second stage juvenile, and they penetrate the root of the host and they go inside and it starts feeding. When they start feeding, they find a suitable place and they form a feeding site called syncytium. And once they start feeding, they don't move and they become sedentary. And after starting feeding, they grow up in size and they become mature and lemon-shaped. This is, I'm showing the female, white female of soybean cyst nematode, but the male retains the vermiform shape and they fertilize the female once they are out of the root. And when they detach from the root and they become dead, they turn into the brown cyst, which contains hundreds of SCN eggs and juveniles. And from the brown cyst, the SCN juvenile hatch under favorable condition, and again, the disease cycle continues. There are a lot of strategies to manage the soybean cyst nematode from the infected field. Obviously, the resistance cultivars are the major strategy, but there are some research that are showing that cover crops can effectively control the soybean cyst nematode from the infected field. And there are 3 strategies that are believed to control the soybean cyst nematode from the field, and one of them is the biofumigation. Biofumigation is generally done from different crop families, especially Brassica family. I'm showing the picture of flower of radish, and they control the soybean cyst nematode by the chemical called thiocyanate and isothiocyanate. These chemicals are detrimental to pathogens like this nematode, and SCN has been effectively controlled by biofumigation and has been shown in many researches. Another group of cover crop which stimulate the suicide hatching also control the soybean cyst nematode from the soil. Suicide hatching is the hatching of these eggs, SCN eggs, in the absence of suitable host. And when they hatch and they don't get the suitable host, they are going to die because of the starvation. So endorrhea ryegrass has shown effect on hatching and death of the SCN juvenile due to starvation. And the last group is a trap crop. This crop enhance the hatching of the SCN as well as they allow the penetration of their root by soybean cyst nematode juveniles, but further, they do not allow the complete development of SCN inside their root. So they are trapping the juvenile inside their root without allowing them to develop further. So our objective was to evaluate 10 cover crops to see their effect on hatching, penetration, and development of SCN, and also to find the effective trap crop from our entries. So first of all, we selected 10 cover crops along with 3 control treatment. 3 control treatment were SCN susceptible soybean cultivar bands. Another was unplanted infested soil, which is a fallow negative control. And another was corn, which is common rotation crop in North Dakota. They had 4 replication and were arranged in completely randomized design. They were planted in growth chamber. We did 2 trials for this research, and first trial had almost 1900, both of the trial had almost 1900 SCN in 1 cone container. We planted 2 sets of plant to harvest 15 days after planting and 30 days after planting to see the change in the population inside the root. And we did the root staining with food coloring dye by using the protocol developed by Thies et al. And the data were analyzed using SAS 9.4 software with ProGlymics and 2KHST to separate the mean. And also we did simple pair t-test to compare the data between 15 days after planting and 30 days after planting. For that, we planted cover crops in a small cone container with infested soil, and then we allowed to grow for 15 and 30 days. Then we extracted the nematode from the soil using the sugar flotation centrifugal method, as well as we separated the roots to stain them for the presence of nematode, and then we observed them under the microscope to quantify the nematode in the soil and root. So this is the first result, first data set obtained from this research. And I'm showing you the data, total juveniles hatched after 15 days of planting. So in the x-axis, you can see cover crops and controls, and in the y-axis, you see the number of hatched juveniles. And if you see here, these 4 crop, including the soybean bands, had the significantly more number of juvenile inside hatched juvenile 15 days after planting than the unplanted infested soil, which means they enhance the hatching of soybean cyst nematode eggs and significantly enhanced than the unplanted infested soil. And soybean cultivar bands are supposed to enhance the nematode hatching because they are susceptible plant. And among the cover crops, we have fava bean cultivar petit, which had the significantly greater hatching rate than all other cover crops, and which is statistically similar to soybean cultivar bands. So it shows that in both trials, fava bean cultivar had the greatest hatching of SCN 15 days after planting amongst the tested cover crop. So in the second set of data, I'm showing you the number of SCN inside the root 15 days after planting and 30 days after planting. This was obtained from the root staining. And in the x-axis, we have the treatments, cover crop and controls. In the y-axis, we have the SCN inside the root. And as you can see from the bar graph, we have fava bean cultivar petit with the highest number of SCN inside the root 15 days after planting. So this is greater than susceptible soybean cultivar bands as well. And if we compare the data from 15 days after planting and 30 days after planting, we see significant difference between them. And most of the cover crop and control had significant difference. But if you look at the fava bean, the number 30 days after planting was significantly lower than 15 days after planting. But in the soybean, the number increased significantly 30 days after planting compared to 15 days after planting. That shows the number of juvenile inside the root of fava bean is decreasing. And we found complete development of white females in the root of soybean as expected, and also in the chorny purple top. The number is small, but still we got the complete development of SCN. In the second trial, we saw similar trend in the total number of SCN penetrated inside the root. We still see fava bean cultivar with the highest number of SCN 15 days after planting, and significantly lower number 30 days after planting as compared to 15 days after planting. Similar trend as in the first trial. Also, we found the formation of white females in soybean cultivar bands and turnip purple top as we found in the first trial. So this data, these numerical data are showing that fava bean enhanced the penetration as well as the hatching in 15 days of the planting. So these visual data, the images from root staining, also support the numerical data that I showed you. This here shows the top image is the juvenile inside the root of fava bean 15 days after planting, whereas this is the juveniles inside the root 15 days after planting in soybean. So it seems like both of the root is providing food to the SCN juvenile because we see both of them are swollen. But when you see the staining image 30 days after planting, the difference is in the soybean, the SCN turned into the complete lemon-shaped white females. But in the root of fava bean, we did not see that development. Instead, we saw the juvenile are kind of degenerating because we did not see, the dense internal body substances when we see the staining image. That means the juvenile are not growing inside the root of fava bean. So from the numerical data and from the staining of the root, we can conclude that significant greater SCN hatching occurred in fava bean as well as significantly greater SCN penetrated the roots 15 days after planting, but those SCN juvenile did not complete their life cycle 30 days after planting. And only soybean and turnip supported the development of the soybean cyst nematode. And from this, we can conclude that fava bean cultivar petit has the potential to act as a trap crop for management of soybean cyst nematode. With that, I would like to thank you for your attention, and I will take any question if you have. Thank you.
Any questions? Yes.
Yeah.
So you tested one genotype from fava beans.
Right, one cultivar.
There's difference in soybeans in their genotypes, how they respond. So is there likely that there is differences in the fava beans in their responses?
Well, since now we have only tested one cultivar of fava bean, but from our previous research, our former lab member also tested another cultivar of fava bean, which is a different type. It's a vegetable type. And also in that type as well, we did not see the reproduction of SCN. From this one, we did on one SG type, but at that time, the research was conducted in 2 SG type, and we did not see the reproduction of both of the soybean cyst nematode on both cultivars. So we cannot really say that all the cultivars or the genotype have the same response against the soybean cyst nematode, but from these 2 cultivars, we only have the